Okay, so this is Michael Hall with Pablo Fabian, and is it Fab? Hold on. You see, this is exactly why we need the dry run.
You're
Is it
fine.
Fabian or
You are good.
okay?
Fabian, yeah.
All right, Fabian and Joseph Headley. We are doing pathophysiology and treatment of COVID-19. I will be focusing on the pathophysiology of COVID-19 specifically. So this is the physiology of the virus COVID-19. Starting from the top left, we have the N protein, which is the nucleocapsid protein. That is what basically holds it binds with the RNA within. Next, clockwise, is the M protein. They call it the matrix protein, but it can also be called the membrane protein. And that has a function with binding to the host's membrane and forming an anchor. The spike protein is these yellow protrusions out of the virus that are used to bind to receptors using a receptor binding domain. The bluish spikes are the envelope protein, which play a role in forming an ion channel. And inside is the single-stranded RNA. Here is the sequence that makes up this virus, and specifically what needs to be pointed out are these structural proteins that are labeled as S, E, M, and N, which is again spike, envelope, membrane, and nucleocapsid. So the first one that I will talk about a little more in detail is the envelope small membrane protein pentamer on the open reading frame 4. And there's two major thought processes on how it binds, how it interacts with the host membrane. The more extensively studied transmembrane domain, which forms a ion channel, as well as on the C-terminal, where it does a type of helical uptake. And those two combined is how the RNA and the virus are transferred into the cell. The membrane protein, which is how the actual virus is anchored onto the membrane of the host cell, helps with the envelope protein forming that transmembrane domain. And it forms a bond with the host membrane and will eventually go on to assist the RNA packaging within, and even further, the death of the cell in apoptosis. The nucleocapsid protein of ORF9A has a role in quite a few processes that are undergone upon the RNA's entrance into our cells, which are replication, a type of transcription, and assembling. The N-terminal of this protein binds to the RNA, and the C-terminal acts as a dimerization inducer, kind of acting to compact that RNA within the virus. And more specifically, even then, those proteins, this focuses a lot on the spike protein because of a lot of our vaccines are being used to target the spike protein, specifically due to its ability to bind to our ACE2 receptors on our cells using the receptor binding domain. You can see it at the top left of the spike protein. And to the right, you can see the actual RBD binding to the ACE2 receptor. I have a real quick figure for the infection, how the infection process moves throughout your cell. And a couple things I really want to point out here are there's the subgenomic transcription. That is what was talked about with the nucleocapsid protein. And a couple of the other parts, like the spike protein finding the the receptor on our cells, and that begins the entire like conformational changes for the membrane and envelope proteins to start infecting. There are a couple of short-term symptoms that are maybe not always shown, but sometimes shown, and you can get any number of variations on this. And there's a good figure on, I guess, a localization of these different symptoms with headaches, fevers, sudden loss of taste or smell, coughs, sore throats, chills with or without shivering, difficulties with breathing, and muscle pain. And then they have a couple that are emergency warning signs that you should be looking to get medical help when you start to experience them, such as trouble breathing, persistent pain or pressure in the chest, sudden confusions, and bluish coloring to your face or lips. This leads into a couple interesting articles that I had found that were really looking at the diverse severities, whereas some people would have COVID and not experience many or any symptoms, and then other people would be very, very deadly symptoms, very severe symptoms. So they were doing a transcriptomics research of people who had very low, not mild symptoms, and where other people had severe symptoms. They were doing an nasopharyngeal swab, and they sequenced these out so they can get a good sequence and find out what is actually happening inside your body. And they found that there is gene regulation, and this is not something that's shocking by any means. Many other viruses have gene regulation, just maybe not to this specific severity, where people who had very mild symptoms had very low gene regulation, and people who had severe symptoms were having greater gene regulation caused by the virus. And this was just downregulating immune productions and upregulating different processes that would further increase the ability of the virus to spread. And then medium-term symptoms that was very interesting to me, where they were looking into people who had gotten COVID, went to the hospital, and then were released, and they just checked with them over a period of about 4 to 6 months. And they did an MRI, and that showed that there was irregular inflammations of different organs, specifically organs that had ACE2 receptors. And this was in correlation with a couple of things that they checked with exercise testing and surveys that showed that there were people who had medium-term effects on their lung capacity with very low likelihood of it ever increasing again. Occult neurological injury where there was inflammation in their heads, like in their brains, that caused multiple injuries and caused them to maybe lose capacity to do certain things or never really recover in the way that you would expect with somebody who had like a concussion or something like that. And then, of course, depleted exercise tolerance with inflammation from lungs, inflammation from a heart. It makes things like low exercise to be much more difficult. And in general, there is a lowered quality of life that was determined by a survey, where people in a control versus people who had COVID and medium-term symptoms would determine whether they had whether they had to answer a bunch of questions, and that would determine how their physical role or their emotional roles were by comparison to somebody who had not had COVID. And it was much lower on specifically those ones and social functioning. And then another questions about their depression and anxiety. And for the most part, there was minimal for those, but when it came to mild to moderate, there was a higher proportion than what would be in a control. And these are my references. So if you guys have any questions.
I do have a question, Michael.
Okay.
Real quick. So you talked about short-term and medium-term. So there's obviously no long-term symptoms just because it's so new.
Right.
Do they have any kind of guesses as to long-term symptoms, what those might look like? Is that something that is responsible to even take a guess at?
Their guesses were kind of in line with what the medium-term symptoms were. They probably could have at some point.
Like a continuation?
Yes. They don't know that these inflammations are ever going to heal fully, especially as you're getting older.
Yeah, that's scary.
It's harder for them to completely heal again. You only get one brain, you only get one heart. I mean, so if there's a lot of these little damages all over it because of the inflammation
that don't heal,
it might not ever go back.
Awesome. Well, that's not awesome, but that was great, Michael.
Thank you.
And it sets me up for my section really well.
All right, I'm going to stop sharing.
I have another question.
Oh, yes, go ahead.
I'll go ahead and share while you're doing that.
I wanted to ask you about the open reading frame, and if you found that there was other sections of genes, certain genes that encoded for something that could possibly be some sort of target besides the spike 1 gene.
I know there was quite a few, specifically on the envelope protein originally, that they wanted to, because even if the spike protein was able to bind to the receptor, the spike didn't really cause the virus to infect by any means. The envelope plays the major role in actually creating this ion channel. So I know a lot of people were originally looking at this a few months back before they started making their vaccines for the spike, of how they could target specifically that envelope protein and attack it, making it ineffective. But I think eventually they found that targeting these spikes are much easier. And if it never binds to the receptor, then it would be unable to infect us and bind with the envelope anyways. But besides that, I don't know anything about like the membrane or the nucleocapsid, because the nucleocapsid so far in, I mean, you've already been basically invaded by the time nucleocapsid is actually doing anything.
It's not a great target.
Yeah, and it's inside of the virus. So.
Hey, unlimited minutes.
Okay,
Did that answer your question?
I have another question about.
Okay. Yes.
Do you know? I have no idea, but I'm not exactly too sure how to ask it, but the epithelial cells on the lung epithelial cells, do they have? They can't heal the way that other cells heal. Like I know for a fact they can't heal like bone. So it's just basically a lot of scar tissue that's formed for the you said mid-long, well,
on the midterm.
Yeah, midterm effects.
Well, and maybe if we're all younger, and if everyone who got it was very young, I think that maybe young people could eventually heal on their lungs. But our organs do have troubles with these. I mean, it's a pretty impressive inflammation of these sites, these organs, and they do not seem to be healing. 4 months should be enough time to heal if it weren't for the fact that it wasn't healing enough. They're still inflamed at 4 months. They're larger in size. So that's danger. That's damage that is not getting healed.
Oh, okay. Yeah, because I know like some cells, most they can be permanent whenever you lose them. I like muscle cells, you can't really regenerate those.
Right.
Once they're gone, they're gone. Or neurons as well, you know. But that's why I was just asking, and thank you so much.
Thank you.
Yeah, Michael, that was great. Okay, so I'll go ahead and start with my transmission of SARS-CoV-2. There's not a lot of cell or molecular biology. It's more looked at a population level, and sometimes like the physics of how stuff is spread. But I'm going to focus. It's going to be pretty short, but I'm going to focus mainly on like distilling these papers into some evidence-based takeaways that you can use, or recommendations for society as a whole. And then I included a couple graphs that I thought were really interesting. They were they were kind of exotic. I'd never seen graphs like that, so I just wanted to highlight those because they had a lot of really good information in a way that I'd never seen it presented. Okay, so right off the bat, we're going to talk about the ACE2 receptors that Michael was talking about. They're everywhere, not everywhere, but they're in a bunch of different cell types in our body. And notably, they're in the gut and the lung tissue. And it's spread predominantly through respiratory. So, and we're going to talk about the difference between droplets and aerosol, what that really means. But transmission is really complex, complicated thing. It depends on the environment. It depends on the virus. It depends on the host. It depends a lot on random chance. But how this specific virus works in humans is that it affects this ACE2 signal receptor, which is part of the angiotensin signaling cascade. And it starts in the lungs. That's why everyone's hearing about lungs and ACE2. But because ACE2 these receptors are found throughout the body, it can infect other tissue types, which is causing a lot of the symptoms, short and medium-term symptoms that Michael was talking about, including, which I thought this one was interesting, human spermatozoa, which we may talk about in a little bit. So right off the bat, the environment is so important for viral transmission. I mean, trillions is not nearly high enough number of viral particles, not just COVID, but viral particles all throughout the world. And so it's everywhere. But it turns out, according to this study by Merowitz, that the virus being out in the world is not how we get sick. It's just not the case. I mean, they can detect viral RNA in the environment, and some studies have polled, like they've, I guess, sort of cultured viable virus. Culture is probably not the right word, isolated. But that's just not how people get sick. Your body has so many natural defenses against viruses, RNases, just how your skin is structured and set up. Like, I mean, you could pick up COVID and like rub it all over yourself and you probably wouldn't get sick because it has to bind to these lung cells. And the way that happens for this particular virus is through droplets. And so if you can see my mouse, you can see how big a droplet is relative to a piece of human hair. So these are substantial. It's not like an aerosol can like Febreze or something. It's larger, almost like spit droplets, and so they stay in the air for less time but can be projected further. And there's a couple other routes, like sexual transmission of viruses. We know that exists in the world, bloodborne, things like Ebola, but that's just, though it's technically possible, it's just so much less common than you're out talking to somebody. Though there is one cluster of fecal respiratory transmission, which I thought was interesting. All right, so temperature. So temperature is really important. Flu season, right? Flu virus is around all the time, but it gets bad when it gets cold out. That's been our experience in this influenza or this virus is similar. So very complex statistical study. I don't put a lot of faith in statistical studies, but transmission in the first 12 days of countries of a certain GDP, and it measures this. You see all this variance in the scatter plot. I mean, it's all over the place, but this line of best fit is clearly going down. So this alpha exponent, this measure of infectivity, with a linear line of best fit, shows that as the temperature gets colder, the virus spreads better. Now, they analyze the exact same data. You can see the dots are all in the same place in a slightly different way. And they got this quadratic line of best fit. And it actually shows that peak infectivity is somewhere in the like 7 to 9 degrees Celsius temperature range. And once it gets to a certain point, it doesn't spread as well. But I don't put a ton of faith in statistical analysis like this because there's just so many different variables. But the main takeaway is that, you know, you don't respond to number of cases. We should start implementing, you know, getting tighter on social distancing and wearing face masks when it starts getting cold, not once cases have started picking back up, because we know why cases are going to pick back up, and that's because of the temperature. All right, so we're going to talk, the last little bit I have is vertical and horizontal transmission. So vertical transmission is going to be mother to child, I think typically. And plus, in the placenta, there are ACE2 receptors. So COVID can be in these placental cells. And I mean, that could potentially be really, really bad. I mean, a similar virus, not similar, but Zika had really severe pregnancy complications. And that could very possibly have been the reality of COVID. But it turns out that that is not the case, though it is present. You can see this brown staining for COVID under the umbilical cord, in the middle, kind of around the edges, and then E is just zoomed in. There's a lot. I mean, COVID is all throughout. You can compare that to this cytocarinin. It's just sort of all over the place. So it's kind of a trade-off. It's like, if you're pregnant and you get COVID, it's less likely to get a severe case. So in some ways, you're protected a little bit. However, if you're one of the unfortunate few who does get severe COVID, it's going to be significantly worse. So it's like, you're better until you're not. And then it's really bad. Yeah, okay. So now we're going to go back to talking about horizontal transmission, person to person. The most recent estimates, this is from 2021, is over half of all transmissions were from asymptomatic individuals. So this nice, like, sunburst looking graph shows us kind of percent infectivity or percent transmission relative to the time between when your symptoms, when you have an onset of symptoms and when you're infectious. So common knowledge might tell us that those are the exact same time. I feel the worst, so I'm the most infectious, but that's just not the case. And as you can see, the people who are peak infectious before their symptoms show up. So asymptomatic people never have symptom onset, but people who are asymptomatic or pre-symptomatic account for so much more, up to like 75% of them will account for 20% of the transmission. Oh my, it's like Mad Max out here. I'm so sorry. All right, and then this is sort of what we were talking about. This is not like a one person infects three people, infects three people, infects three people kind of virus. It just doesn't work like that. Most people infect no one, and some people infect, a few people infect a lot of people each. And those are the super spreading events that we've heard about. And so here is another interesting chart talking about days to symptom onset versus, this is like infectiousness versus when you start like having symptoms. And so people are actually more infectious, much more infectious before they have symptoms. And so that's why it's spreading so badly, because once people are sick, they quarantine, but the virus is still spreading like crazy. And so this is my last graph. I thought this one was really interesting. This is broken up into 3 sets. It did contact tracing for 100 clusters of people. And so the idea is this green bar is going to be when you were exposed to another person. And so, I'm sorry, is when you're, sorry, there's 2 people. There's your primary case, which is the red, and then there's the secondary case, which is the blue. These are when your symptoms start, and the green is when they interacted with each other. So here you can see the green is before the first person experiences symptoms. Here you can see the first person experiences symptoms while they're interacting with each other. And then here you can see they don't interact until after the person has experienced their symptoms. And so the main takeaway from this is that contact tracing needs to happen way before, because it's not it's not including all of this pre-symptomatic transmission. So the big takeaways from this are just that all those preventative measures that we've been doing are so important, and they're maybe even more important than we thought, because up to 5 days before you even start feeling sick, you can be just giving it out to everybody. All right, and so I focused in on about 6 of my references, so I didn't use all 10. Do y'all have any questions for me?
I absolutely have some questions.
Hey.
Okay. I have 2 statements, I guess. First one, have you heard about New Zealand? It kind of goes into your contact tracing point, where they just got one person in one of their major cities just recently that had a different strain of COVID. So they just were like, okay, the whole city shut down for 7 days. We don't know if you came in contact with anyone, so we're just going to shut the whole thing down.
Yeah.
And they just immediately are like, nope, we're going right into all the precautions that we can, because we're going to shut it down immediately.
I had not heard about that, but that's
I can't remember the city.
In some ways it sounds extreme, but in others, I mean, yeah, the seven days where you all shut down, but if no one else gets it, I mean, it could drag on for weeks or months or years.
Well, 7 days, I think, is just to, I guess they're trying to root out who you came in contact with and anyone else, so that they can, easy contact tracing with that.
Yeah.
And I think that's very smart. I mean, it's a little easier to do it there than here, but it is still so important. The second statement is, I really like the asymptomatic chart right there. I like that. That was very, very useful.
I struggled to read it at first because it has a lot of information. I mean,
yes, it does.
Two axes. It's got these strange like bands, and it tries to draw a distinction between pre-symptomatic and asymptomatic, which can be a difficult kind of nuance or like line to walk. But yeah, I thought that was really interesting that and you can see this gradual increase towards people that, like, almost all of these infections are from people that are pre-symptomatic by more than 2 days. So like they're peak infectious 2 days before their symptoms. And so
basically it's just that they don't know that they have it, so they're going out and doing things, and that's causing more and more people to catch it. Is that basically it?
Yeah, and they're not wearing masks, and they're not social distancing and whatever. And so for those reasons, they are doing this heterogeneous transmission, right? They're given, one person may infect 10 or 100 people easily. Whereas, and I think that kind of ties into host like biochemistry and physiology, but that's not what I really focus on for this one.
All right. I mean, I didn't ask my question, but you kind of already answered it. But thank you.
What's that?
It was basically, so I kind of was just leading into what I was going to ask, but then I kind of like answered myself a little. Where I was like,
great.
okay. So basically it's spreading mostly or a lot because of people just asymptomatic. And that is having a higher rate of spreading, right?
Sorry, say that one more time. It's spreading because people
it's
are
spreading as bad
asymptomatic?
as it is because people who are asymptomatic don't know that they have it, and they're just going out and doing things.
I would think the vast majority of people, once they're experiencing symptoms, I mean, everyone stays home when they're sick generally, and if they're very sick, you would think that they couldn't not stay home unless they're a real trooper. But yeah, it's spreading because people are asymptomatic or pre-symptomatic, and then not also following the public health guidelines that are clearly established and that everyone should follow voluntarily. But yeah.
Yes. So, well, thank you.
All right. So I will stop sharing. Pablo, you ready?
Yes, I'm ready.
Hey.
Oh wait, let me see something real fast. All right, can y'all hear me? Just making sure. All right,
yes.
So everybody knows my name is Pablo Fabian, and I'm doing the treatment for COVID-19. So I want to briefly just go over the treatment. We've already talked about the transmission, and the treatment consists of repurposed drugs, the convalescent plasma, immunoglobulins, stem cells, and the vaccines, the ones that are being very popular right now. The most one being the RNA vaccine, but we also have protein subunit and active virus vaccines and adenovirus as well. So I'm gonna try to go as fast as I can and try to summarize some things. It's just a lot of information, but let's get started. So the first one I want to talk about is the subunit vaccine. So basically what it is, it's incorporating a piece of the pathogen into the vaccine. In this case, most of these that have incorporated something, it's a spike 1 protein. And right now they're looking a lot into the actual vehicle that delivers that. So the main ones that I found were the liposome and virusome, although there is a polymeric nanoparticles. So how this actually works is, so it's embedded into the liposome, and the liposome actually goes through the membrane of the cell and delivers the actual protein. And as we know before, the way that it actually envelops or actually delivers it is by recognizing that there is a PAMP, as we talked before, those patterns on the pathogen that recognize it as foreign. Once that happens, it comes in, it's modified and processed, and it's then put up on the surface of the cell as the I believe it's major histocompatibility complex. That now then stimulates a response. So there's a lot to actually talk about when it comes to the formulation of these drugs, vaccines. What makes them very important and efficient is that they also carry toll-like receptors. So these receptors also help stimulate an immune response, and they're actually a lot more efficient than the actual ones that carry, or traditional that carry aluminum adjuvants. Adjuvants are just another word for the delivery vehicle. So the ones that are actually doing the research are the Baylor College of Medicine, So they basically have formulated a vaccine that has the S protein binding domain, and they also have toll-like receptors. So basically for these, like the other University of Queensland and Novavax, Clover Biopharmaceuticals, and GlaxoSmithKline, they all have a sort of a similarity in the way that they're doing their vaccines. Their signature products is basically made on the vehicle and what they have besides the spike one protein or the domain. So that's what it mainly is. For some of these, they've actually gone into clinical trials. The Novavax vaccine is currently on phase 3. And this is just a paper that was published in January of this year. So that's just what that paper had let us know. And for inactive virus vaccines, as we know, they have where it's either the whole virus, but it's not alive anymore. It's dead by physical or chemical processes. And for this, they actually used the whole drug, the whole virus. The one that actually first started was the Yuan Institute of Biological Products, and they actually tried to culture this into, I believe it's a sort of rhesus mosquitoes. So they've actually done that on animals, and now they're on phase 3, as of that paper had confirmed. Another one is the University of Wisconsin, and they actually collaborate with Flugen and Biotech, and they actually are doing something very similar, but their vaccine is actually intranasal, so it's not a traditional intravenous vaccine that carries the inactive virus. All right, and let's go and see what's next. Adenovirus. So the adenovirus is just it's used as a vehicle as well. Well, more like a vector, to be very exact. And what it has this is the actual definition: vaccine that has a double-stranded linear DNA enveloped into the capsid, but it does actually contain the DNA. I mean, not the DNA. I'm sorry, the RNA of the virus embedded into the adenovirus. So they actually have this, and what it traditionally does is it also causes an inflammatory cytokines, which later should stimulate an immune response. And it goes inside. The mechanism is very similar to a traditional virus, and once inside, the vector hacks the same molecular system and produces spike protein. So most of these actually have a very similar process where they have to bring in the virus particle or whatnot, and it's processed where it can be then shown on its surface and then stimulate an immune response, and have T cell immunity. All right, but now we look at the DNA vaccines and RNA vaccines. And I forgot to mention the Oxford University Jenner Institute and CanSino Biogenics are actually the ones doing the adenovirus research. So I believe that they're actually in phase 2. The CanSino Biogenics is in phase 2. And that's was mentioned in the paper. Let me go back to the DNA RNA vaccines. So the DNA vaccines are actually embedded into a plasmid. So that's how they are then taken up by the cell, and it goes through the nucleus in order to produce the protein of interest, which is spike 1 again. For the RNA vaccine, so there's actually, and this one's the most popular one, the RNA vaccine is basically messenger RNA, and it's also embedded into a liposome, so it's very it's very easily crossed through the membrane, and it actually is processed like any other mRNA in the cytosol, thus producing the spike 1 protein. And then once the spike 1 protein is gone through the all of the translational process and gone outside of the and the extracellular plasma outside the cell, it is then, that's when it actually produces the same response, and it's actually brought in again and actually processed as a foreign entity at that point. Another thing we have to see is that the Inuvo pharmaceutical, the one that is actually doing the DNA plasmid vaccine, is in phase 2. That's what it mentions in the paper. For the other ones that were mentioned were Moderna, and that one's actually, it says phase 3 clinical trials, but as we know, like, they're using it now. So, and also the Pfizer one that's we also know about, and that one also says it's on phase 3, but as we know, it's already out. So that's that. So there's antiviral medications, and I'm gonna just try to briefly touch on these. There is RNA-dependent RNase polymerase inhibitors. What these actually do is they actually bind to the mRNA template, thus not allowing the RNase polymerase to attach, and there isn't any transcription that occurs. Any transcription that occurs. So, and there's also protease inhibitors, and the protease inhibitors are used to inhibit protease that are actually expressed by the open reading frame. The open reading frame not only encodes for from what I know, it does not only encode for the spike 1, but also it encodes for non-structural proteins. And these, I believe there is 16, but I could be wrong. It could be less than that. But what these do is they actually later on help construct the replicase that is used to replicate the actual virus. So once targeting these protease, there is no replicase that will occur or come to be. There's also, and that's lopinavir and ritonavir. As we know now, they're actually not very efficient and clinical trials, because all these repurposed drugs, they have to go into clinical trials again. And that has to do whether the actual indication is different, the route of administration is different. So they're not exempt from clinical trials. They have to do that. Because the RNA-dependent RNase polymerase inhibitors were used for HIV, and so were the protease inhibitors. There's also something called the infusion inhibitors. So basically what they do is they just bind to the spike 1 protein. It's almost like having a balloon or having a ball full of spikes, and then just having a lot of marshmallows on those spikes, so it really doesn't hurt anything or anybody. And so it never really infuses into the membrane to then process all those proteins that it has for replication and whatnot. There's anti-malarial drugs as well. So these anti-malarial drugs, so they inhibit the replication by increasing the endosomal vesicle pH. So and these are chloroquine and hydrochloroquine. So there was an idea there that these drugs could inhibit the proteases that are involved in post-translation. As we know, that there is a process in which the viral proteins are made and formed, and one of the last steps is post-translational. As we know, if that's inhibited, there's no use for these for these proteins, or they won't assemble the way they should. And so as we know, they're not very useful. They haven't showed efficacy. There also is angiotensin receptor blockers and angiotensin converting enzyme inhibitors. So as we know now, there is a big controversial debate whether they should be used or not. There's actually been research that shows that they do not cause, they do not contribute to the infection or any severe symptoms. And on the contrary, they actually they help out with those symptoms. And it says there that research found that 19 treated with ACE inhibitors were 40% likely to be hospitalized than those that were not treated. Another thing that is important to take in is that the ACE inhibitors, they actually allow the expression of ACE2, and because of that, they could actually express cytokines, but it also, I think it actually depends on how severe the case is, because if there is cytokine storm, that can actually cause organ failure, as we talked before. So there's still a huge debate how they actually, or who they actually work best for. This is just a mechanism of action. So as we see the names of the actual of those drugs, repurposed drugs, and we can see where they actually may inhibit a certain process. So this is basically just a way that you can picture what I just said, and we can move on to the next ones. All right. So novel therapy is mainly based on biologics, and we can see that all these are they are very similar. So the first ones I want to talk about are the inflammatory modulators. So monoclonal antibody therapy is used for the side effects, you could say, or to actually combat something that's causing a major side effect of the infection. For these two, I will definitely mess up saying those. So let's just call them T and E. T drug is actually used, and it's been there's been a study conducted in Yuan, China, and they confirmed that it blocks the interleukin 6 receptors. Because of the blockage of the receptors, it's actually downregulated cytokines, and it actually improved the clinical improvement in patients that had severe COVID. So that's that's been helpful. That's just some results that have came to mind, and I wanted just to put out there. So the E-drug is also a monoclonal antibody that binds to complement component 5. This actually it actually blocks a pathway that later stimulates cytokine release. You also have the interferon alpha and beta, which are, as we know, a family of proteins that have an anti-inflammatory property that downregulate the production of pro-inflammatory cytokines. And so that's just the beta, and the alpha actually activates T cell responses. So we can see that there's, different kind of strategies throughout finding what we could use for this disease. There are also stem cells that have been hypothesized uh, that could be of some use. They mentioned the mesenchymal stem cells a lot, and that they could actually help people recover. And that's why I was asking before whether the lung damage was permanent or not, because stem cells can actually regenerate these epithelial lung cells, and it also has other properties. It can downregulate cytokine, um, and also, what else? I know it had some other property. It also has another property with the regulation of T and B cells. Um, and although that the mesenchymal stem cells are mentioned here as umbilical cord from that source, they can also be isolated from bone marrow and peripheral blood samples, adipose tissue. So it doesn't necessarily need to be from that source. And another one I want to talk about is the convalescent plasma treatment. As we know, this is one of the first treatments that was available to people. As people recovered, they just wanted to actually get the plasma because they were hoping that it had the immunoglobulins to combat the virus. So basically, they were just doing a transfusion. And although it helped somewhat, it is not the first line therapy or first choice that people would use. And the last one is immunoglobulins. That's more of a extraction and purification of the convalescent plasma treatment. Although they not necessarily are from people, sometimes they are humanized from mice, and where they actually target single epitopes, and that's what makes it more highly specific. Those are the research institutions, Lilly Pharma, um, and the Regneron, that have actually looked into it. These immunoglobulins are actually very helpful. And this one's more of a direct towards the virus, not like the ones that were before, where it has inflammatory modulators that are not so focused on the virus itself. It's more of a peripheral thing, even though it does help. So there's also non-pharmacological treatments, and I separated them on invasive treatment and non-invasive. So there's 3 that were mentioned, and they were mentioned on a paper that had to do. It was just basically a review of what was out there that was non-drug. And this is the ones they listed: the high flow nasal cannula. So basically, it's recommended for people that have acute respiratory distress syndrome. And what it is, it's actually it's heated and modified modified air, and it has a number of benefits. So basically, it's just giving warm, soothing air instead of dry and cold that could actually, lead to airway inflammation. Um, so, and it has helped people decrease, like I said, inflammation, um, and actually decrease in energy expenditure. So they're not really so tired and exhausted. Um, and there's also the extracorporeal membrane oxygenation. So it is basically a way to replace cardiopulmonary support. What it does is just drains blood from the vascular system with a pump. Um, and basically, this is just a way of gas exchange from O2, from CO2 to O2, and thus helping the patient out. It's more used for severe cases of acute respiratory distress syndrome, and those that are actually, you know, um, at high risk of death. There's also the high frequency oscillator ventilation. The treatment depends on the baseline severity of hypoxia and damage to patient. Um, but basically, it's just a respiratory therapy at high rates of exchange of gas exchange. Um, and there's also non-invasive therapy. The prone position, just basically lying face down, and most of the time you have your face down on somewhat like a bed, um, like the ones that they actually have for massages. And most of the people that have laid on these at a hospital was because the beds are just full. Um, and they found out that even though these beds are full and all these equipment are being used, that they can have a prone position, it may reduce the severity and actually have them breathe a bit better. Um, and there's also inhaled nitric oxide. So they believe that the nitric oxide can dilate blood vessels. Um, but the author of this study concluded that it doesn't show a significant improvement. Um, and basically, this is it. There is a lot more. These are just some pictures, just so you can get a visual on these non-pharmacological therapy uses. And these are just oxygen therapy. That's all they are really. And that is it. Those are my references. And do y'all have any questions? I know that was a lot, but there is so much more information out there, um, and more detailed information. But I hope this actually helped out.
Yeah, that was great. Can you go back a couple slides? Let me see. So I was just talking to my uncle about. I was thinking about this when you were talking about the vaccines.
Yeah.
And there's all these great medicines, and especially the vaccines, and he's just not going to take it because he's crazy. And I don't know. I just want to vent about that because he was trying to tell me the vaccines can kill people. He's like, all these people have died. He doesn't get any kind of good data from anything. But have you seen in all your stuff looking at this? Have you seen anything about a long-term side effect from a vaccine at all?
From the studies, no, I have not seen a long-term. I mean, the short-term, well, basically just like a few days.
Yeah,
You may get sore, you might
it's
have aches.
it's
You might have,
mRNA. It's not.
yeah.
It breaks down so fast. But so yeah, you didn't see anything specific about that. And
No.
then all of these. So I'm seeing a lot of like repurposing of drugs that already exist. These things that end with MAB, these monoclonal antibodies, and are they developing a COVID specific drug? Like you talked some about the immunoglobulin immunoglobulin therapy. So that would probably be COVID specific, but
Yes, that is.
But are there any? Do you know of anything in the works? Any sort of like, I don't know what word kind of specifies these things over here. The like a drug specifically that's not an immunoglobulin that's specifically targeted for COVID,
I mean,
or is it just other viral things that are being
the ones before are
repurposed?
like repurposed drugs, or just they were used, like I said, for
Mhm.
HIV.
Mhm.
On the previous slide. Oh no, the one before that. Like these are like chemical based. These are just, and they were used for the antiviral were basically used for HIV, and because they have so much similarities, like HIV and SARS-CoV-2,
Mhm.
they're able to use the same same mechanism. But these are direct. These are more like whenever you're already infected. Like the only one that may be helped like to prevent is infusion inhibitor, or that might be actually used to have a lower viral load. But yeah, this is the only one, the infusion inhibitor, the one that maybe very preventive use.
Okay, that's awesome. That's I learned a whole bunch in your presentation. I think drugs are so interesting. My parents used to work for a pharmaceutical company, and so I've thought about stuff like that since I was like 5 years old.
Yeah, I like it too. I want to be able to actually make something later on in the future.
Yeah, and you contribute to one drug that could save
Yeah.
millions or more potentially.
Like
People.
Chelsea, I think she mentioned something about a peptide, like a synthetic peptide that she was developing with Dr. Banerjee. I think
Mhm.
that was last semester when she had mentioned something. I was like, oh, that's really cool. Um, and there's actually a vaccine that I maybe didn't mention. No, I didn't mention it. But there is peptide vaccines too, where they actually have synthetic peptide, and it may be just based on a certain part of a biological agent that can work against COVID.
Mhm.
Just a small part, basically just where the instead of using the whole thing, just use like that main part that just targets it, that lands on it and inhibits whatever process. So those are like the synthetic ones too. So I mean, there's a whole bunch of ways one can go with designing
Mhm.
a drug.
Yeah. Yeah, that's why. Well, thank you.
Thank
Michael, do you have any like wrap up points or questions or anything?
I don't think so. That was very comprehensive. I wasn't actually sure that there was this much in terms of
Oh,
like different types of
yeah, there's so much.
treatments.
Oh my God, it's very interesting, but it's overwhelming to know too, because you're just like, man, I get it, but I want to know more, and it gets even more complicated.
Mm-hmm. Every day, it seems you read one paper and that tells you how many, you got to read 10 more now.
Yeah,
Exactly.
definitely. I get that.
Uh, but
It's
eventually, we'll probably not eventually read them all, but you'll read enough to be like, yeah, I got this. And you'll
Yeah,
be an expert in like the tiniest thing,
right.
but you'll know
it's
it
good
really,
to
really
know.
well.
It's good to know that there is like there's a future of it though, because with the vaccines that we have now, there's so many questions of whether they're going to be effective against anything else, any mutations, any changes of the virus. And maybe there's an end in sight with all of this research being done on alternative methods.
Mhm.
Right.
Yeah.
And we can also roll out things that are not working. I mean,
Mhm.
that's good too.
Yep.
And we're only going to get faster at making, I mean, if we can make a vaccine in a year and have it distributed to the world, the next time we can probably also do it in a year or maybe even faster, you know? So that is promising for, you know, just the human global health into the future. You know, I just think that that's exciting.
Yeah.
But, all right. Well, thank y'all.
Thank you.
Michael, can you
I think
send it to
everybody did a
me?
great job.
Yeah, I think
Yeah.
so.
Yeah, I think that's a wrap probably.